Hello everyone, myself Sunita Sahoo, student of MSc 3rd Sem. My topic is mitochondrial DNA coded cellular damage. I am going to explain this topic in 3 parts. First, we will see the structure and function of mitochondrial DNA. Second, we will see the reactive oxygen species mediated mitochondrial DNA damage. And finally, the cellular damage and disease related to the mitochondrial DNA. We all know mitochondria is the powerhouse of cell as they produce cellular energy in form of ATP by oxidation of food. We also know that they are known as the semi-autonomous organelles because they have their own DNA. Although most of the DNA is packed in chromosome within the nucleus, mitochondria also have a small amount of their own DNA which is circular and double-stranded. This is known as mitochondrial DNA. Most of the proteins are encoded by the nuclear DNA, but mitochondrial DNA also has some proteins that are very essential for our body. The size of mitochondrial DNA varies among different species. In humans, the mitochondrial DNA is about 16,500 base pairs long. It is said that mitochondrial DNA is similar to the bacterial plasmid. Mitochondrial DNA contains 37 genes, all of which are essential for the normal mitochondrial functions. Out of these 37 genes, 13 of these genes provide instructions for making enzymes involved in the oxidative phosphorylation or the electron transport chain. These 13 genes encode 7 subunits of complex 1 of electron transport chain that is known as NADH dehydrogenase, 1 subunit of complex 3 that is cytochrome BC1, 3 subunits of complex 4 that is cytochrome oxidase, and 2 subunits of complex 5 that is ATP synthetase. Now let's understand the functions of the mitochondrial DNA. So mitochondrial DNA encodes small amount of proteins that are involved in proper working and regulation of the electron transport chain to generate ATP. The second function is regulation of innate immunity. We all know that innate immunity is the inborn system that recognizes and responds to infections by pathogen, providing immediate and non-specific defense. So in innate immunity, the mitochondrial antiviral signaling proteins, that is the MAPs, play a key role in the innate response to viral infections, helping to induce antiviral and anti-inflammatory pathways. The third function is programmed cell death, which is also known as apoptosis. So apoptosis is the highly controlled process of programmed cell death, which is used by multicellular organisms in a number of biological processes. Mitochondria control the intrinsic pathway of apoptosis. It releases proteins such as cytochrome C from their intermembrane space in response to cell stress. These cell stress can be heat or any infection. Disturbances to these regulations are associated with the development of tissue damage, cancer, etc. The fourth function is stem cell regulation. We all know that stem cells have property of differentiation and redifferentiation. So mitochondria are thought to play crucial roles in the maintenance of pluripotency, differentiation and reprogramming of the induced stem cells. So now let's move to the second part, that is the reactive oxygen species mediated mitochondrial DNA damage. So the sources of mitochondrial DNA damage can be endogenous, cytoplasmic or any environmental source. The persistence of mitochondrial DNA damage can cause mitochondrial degradation. Faulty repair or replication can result in mitochondrial DNA point mutation or deletion, leading to loss of mitochondrial DNA function. Mitochondrial membrane depolarization and increased reactive oxygen species production in the mitochondrial fragments triggers cell destruction by mitophagy. Increased reactive oxygen species or loss of DNA repair enzymes can increase mitochondrial damage and mitophagy of dysfunctional mitochondrial fragments. Impaired mitochondrial function is a hallmark of aging and is involved in a large number of disorders including the neurodegenerative disease such as the Parkinson's disease, diabetes, cancer, autoimmunity. Oxidants cause more mitochondrial DNA damage than nuclear DNA damage. Reactive oxidant species can damage various cellular components such as proteins, lipids, and DNA. They can damage mitochondrial macromolecules either at or near the site of formation. Therefore, in addition to the role of mitochondria as a source of reactive oxygen species, the mitochondria themselves can be damaged by reactive oxygen species. Mitochondrial injury is reflected by mitochondrial DNA damage as well as by decline in the mitochondrial RNA transcripts, protein synthesis, and mitochondrial function. Mitochondrial DNA is more susceptible to oxidative attack than nuclear DNA because of its proximity to the respiratory chain and its poor repair activity against damage. Mitochondrial reactive oxygen species may result in the progressive destruction of the mitochondrial DNA. Such mitochondrial DNA damage leads to the loss of function. Mitochondrial DNA is highly susceptible to damage by hydrogen peroxide. Empty DNA damage leads to apoptosis or inflammation. Mitochondria is involved in intrinsic apoptotic pathway. Activated caspase are a hallmark of apoptosis induced by the intrinsic pathway. Caspase are the enzymes that are activated not just to kill the cell but to prevent the dying cells from triggering a host immune response. Caspase cascade suppresses type 1 interferon production by cells undergoing BAC or BAX mediated apoptosis. BAC or BAX trigger the release of mitochondrial DNA. This is recognized by C-gas or sting-dependent DNA sensing pathway which initiates the interferon production. Activation of the apoptic caspase cascade blocks this pathway. The second consequence of mitochondrial DNA damage is inflammation. So mitochondrial DNA possesses unique features that are immunostimulatory. Damage to mitochondrial DNA from stressed mitochondria can activate inflammasome that triggers interleukin 1 beta and interleukin 18 production. Mitochondrial DNA also engages the STING pathway to induce interferon production. It activates the TLR9 pathway to induce pro-inflammatory cytokine and chemokine production. Inflammation benefits the host by promoting the elimination of pathogens and clearance of cellular debris. Mitochondrial DNA, the only form of non-nuclear DNA in eukaryotic cells, is a major activator of inflammation. In this way, dysregulated mitochondrial DNA metabolism can promote chronic inflammation and disease progression. Consequences of mitochondrial DNA damage is: first is oxidative damage to mitochondrial proteins and membranes. Second is defective steps involved in Krebs cycle, fatty acid oxidation, urea cycle, and also amino acid metabolism. Third one is the increased release of cytochrome C that leads to activation of apoptosis cascade. Fourth one is the neurodegenerative disease such as the Parkinson's disease. Fifth one is the defect in innate immune response and atherosclerosis. And finally, it leads to cell death, inflammation, and defects in metabolic processes. Reactive oxygen species can cause various types of oxidative damage, including DNA strand breaks, base modification, or removal and cross-linking. For proper functioning of mitochondria, maintenance of mitochondrial DNA is required. Now, maintenance of mitochondrial DNA through replication and repair requires the import of nuclear DNA encoded proteins. Thus, mitochondria completely rely on the nucleus to prevent mitochondrial genetic alterations. There is an important crosstalk between mitochondria and the nucleus that helps in maintenance of mitochondrial DNA. Mitochondrial damage or loss of mitochondrial DNA repair proteins promotes a cascade of quality control events, and defect in these events leads to cellular damage, pathology, and disease. Now I would like to conclude by saying that mitochondria are of central importance in eukaryotic cells. However, mitochondrial DNA is more prone to damage, and mitochondrial DNA repair pathways are inadequate. Together, these problems might frequently lead to unrepaired mitochondrial DNA problems and defective energy metabolism. Mitochondrial DNA damage has been frequently shown to be involved in initiation and progression of several diseases, including various types of neurodegenerative disease, cancer, heart disease, and diabetes. There is an urgent need for detailed investigation in this area to find out the mitochondrial contribution to various diseases so that the improved prevention measures and cures can be developed. Thank you.